In this video, I'm going to briefly walk through the vesicular transport section of your Chapter 3 notes. With vesicular transport, it's named because of vesicles within the cell, which are facilitating each process. The two that we look at are endocytosis and exocytosis. Endo, meaning inside, is vesicular transport of particles into a cell. The vesicle itself will be formed from the plasma membrane, and there's several different processes that are associated with the overarching theme of endocytosis: phagocytosis, pinocytosis, and what's known as receptor-mediated endocytosis. This is an active process, so ATP is required, and what's being transported is two or more solutes moving in the same direction. So in looking at endocytosis itself, again, I gave you the definition: vesicular transport of particles into the cell. Phagocytosis is cell eating. So this is where a cell like a macrophage, phage, named for the process phagocytosis, can actually engulf something that is extracellular and bring it into its cytoplasm. Pinocytosis does the same thing, but instead of looking for larger entities, this tends to bring in small bits of ECF, almost like it's drinking small amounts of fluid. That way it can assess the solute concentration of the fluid that is around it. So with phagocytosis, whenever that occurs, we see a number of things happening to what's been engulfed. So for example, it shows you right over here a phagocytic cell like a macrophage engulfing some foreign particulate matter. It literally begins to pinch that plasma membrane inwards until it completely forms a vesicle around that particulate matter. This original vesicle is called a phagosome because of phagocytosis. Now, there's many things that can be done with this, as you'll see in the next slide, but for right now, I'll focus on what happens if this encounters a lysosome. So this is good review for your organelles. Lysosomes are important for digestion of materials within the lysosome. So what can happen is a phagosome can actually merge with that lysosome because they're both part of the endomembrane system. That's going to be referred to then as a phagolysosome once they've merged, right there. At that point in time, the enzymes, which were originally contained in the lysosome, now have the ability to digest any particulate matter from the phagosome. Once that's occurred, a number of things can happen with the contents of the phagolysosome. In this example, it shows you that that vesicle then once again fuses with that plasma membrane and then allows the digested particulate matter to be eliminated from the cell. This is actually what we know as exocytosis, which we'll look at in a second, and it would be the opposite of endocytosis. When you're looking at receptor-mediated endocytosis, this becomes a more specific kind of endocytosis. What you would see here is receptors which line the extracellular surface of the plasma membrane, and those extracellular molecules would actually bind to the receptor before they can be engulfed and phagocytosed. This is a way of making sure that very specific things are brought into the cell. So if a cell itself is trying to respond to a whole cell in itself, like an immune cell or some kind of a signaling protein, it can make sure that that's built up in a certain quantity and that that's brought in at a certain time. However, we find that viruses, for example, like those associated with a cold sore, can actually look like one of the extracellular molecules made by your body and trick the body into facilitating receptor-mediated endocytosis. As we move on then, we take another look at what happens after endocytosis occurs. The reason I put this slide here is because it actually does show you a clathrin-coated vesicle, much like what you would see in receptor-mediated endocytosis, but this combines the concepts that we see both following phagocytosis and receptor-mediated endocytosis. So once again, as you begin to engulf some kind of a particulate matter or a bacterium or cellular debris, that enters the cell as one of these vesicles, which we called a phagosome earlier. If it happens to be clathrin-coated with those receptors, then that becomes uncoated here, right, within the middle of the process. And those clathrin receptors can actually be recycled and implanted back into the plasma membrane to facilitate more receptor-mediated endocytosis at a later time. Now, that phagosome, which can also begin to merge with a lysosome to form a phagolysosome, could facilitate digestion of that particulate matter or debris. What it could also do is it could allow that material to be tasted and tested or altered and then actually deposited back again here on the other side of the membrane. So this is a transcytosis, where you have transport of something from one side of the cell to the other. And so that just gives you some more examples of what can happen following phagocytosis. Now with exocytosis, we see that this is the opposite of endocytosis. And this is where you're going to have some kind of a secretory vesicle named for the word secretion. And any kind of a secretion is a cellular product that is eliminated from the cell. So this is a secretory vesicle. You're going to find that these bind here to the plasma membrane using some of those membrane proteins that are there. And they actually begin to become part of the plasma membrane as they then allow their secretion to be emptied. And so whenever a cell makes something like a hormone, enzymes, or even lactation glands making milk, this is the way those items are going to be produced and then secreted from the cell. Likewise, when you were reading about organelles like the rough ER and the smooth ER, which have the ability to replace missing components of the plasma membrane, this is how they would do it. They essentially just make a vesicle that can then travel towards the plasma membrane and embed within the plasma membrane to replace missing or damaged parts of the plasma membrane. And that's exocytosis. Here I show you examples of pinocytosis and exocytosis using a YouTube video that you can see here. We actually used this video within lecture to discuss the endomembrane system and how all of your organelles considered part of the endomembrane system can actually bind to and communicate with one another. And so this video just basically summarizes all those processes into one. And I like this image here because you can actually see some of these vesicles within a cell, right? And it gives you that nice rounded shape. And of course, it's showing that some of them are binding with the plasma membrane right across here. Right? Some of them you can even see kind of opening up just like that. And this is showing you exocytosis of some substance into the interstitial fluid between two cells. And so that's vesicular transport, both examples, endocytosis and exocytosis. And hopefully that helps you understand this a little bit more clearly.